listeners of the Bio Report, I want to tell you about a new member benefit from the California Technology Council. CTC has teamed with Reprivata to offer members 6 months of Reprivata's COT network service for free, which gives companies the power of a VPN at a fraction of the cost. A remote, flexible workforce is the new normal, but most corporate networks aren't built to accommodate work from home at scale. Reprivata's COT network offers an easily deployable, affordable, and scalable solution to securely enable remote workers and protect the corporate network. To learn more about this and other member benefits, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. The problem of drug resistance isn't limited to bacteria. It's also a growing concern with fungal species that's causing an increasing need for new agents to combat these microbes. Scynexis is developing the experimental drug ibrexafungerp, the first of a new class of therapies for serious fungal infections. We spoke to Marco Taglietti, president and CEO of Scynexis, about the problem of drug-resistant fungal infections, the company's experimental therapy, ibrexafungerp, and why it may provide a new way of treating a range of serious fungal infections. Marco, thanks for joining us.
Oh, my pleasure, Danny, for inviting me.
We're going to talk about drug-resistant fungal infections, Scynexis, and your efforts to develop antifungals to treat these infections. Maybe we can start with how big a problem resistant fungal infections represent today.
It's a growing problem, and simply because antifungals have been introduced many decades ago, and there are very few of them, only 3 classes. One, the polyenes introduced 60 years ago, the azoles introduced 40 years ago, and the quinocandins introduced 20 years ago. And of course, with all this time, the fungi had an opportunity to develop defenses against these 3 classes. This is why we are developing a new class in order to overcome the growing problem of resistance to antifungals.
Well, how threatening are these infections?
Well, the type of infection that we are treating are difficult to treat and invasive fungal infections. Invasive fungal infections, meaning infection of the bloodstream, of internal organs, usually they happen in patients who are immunocompromised. So cancer patients with undergoing chemotherapy, or transplanted patients, solid organ transplant, bone marrow transplanted patients. And these patients, they don't have the immunodefense to fight these infections. So these infections, they are very aggressive. They can spread inside the body of the patients, and the mortality of these infections can be still now, with the best treatment, between 20 to 50%. 20 to 50%. So extremely high mortality. You have also infections that are not internal, like, for example, mucosal infections of the esophagus, of the mouth, of the vagina, that can be very, very difficult to treat. And these are all the type of infections, invasive and of the mucosa, that we are trying to treat now with our compound.
We hear a lot about antibiotic resistance and the causes of that. Why are we having this problem with resistant fungal infections?
Very good question, Danny. The reason the resistance to antibacterials has been on the front line is because bacteria are very quick in developing resistance. And the reason is because they are very promiscuous. They exchange DNA not only between the same species, but also between different species of bacteria. And therefore, development of resistance to antibacterials usually develops very quickly after a few years of the introduction of the product on the market. And fungal infections and fungi, the development of resistance is slower. It may take 10, 15 years before the significant amount of fungi become resistant. And this is why now we start to see the problem becoming bigger and bigger when you start to see classes that have been introduced 20 or 40 years ago or even longer. And so the reason of the focus on antibacterials is because they develop resistance very quickly, and therefore this has been the focus of the last probably 15, 20 years. But the resistance to antifungals is becoming a major issue, and you have new species emerging, like Candida auris, that has been shown to be multi-drug resistance with high mortality, very difficult to control infections.
Given the limited arsenal for antifungals today, what happens when a patient is resistant? How are they treated?
Very good question. I'm an infectious disease specialist by training, by the way, and I tell you that sometimes what we do as doctors is just to, what is the expression, throwing the sink to the patient. Sometimes we use all the antifungals we have available, all at the same time, in trying to control those infections that can be, as I said, deadly. So, the attempt is to give an antifungal, which one of the three classes available, so an azole, an echinocandin, maybe amphotericin B, all together, hoping that something will stick.
There have been some efforts to encourage the development of new antibiotics through such measures as the GAIN Act. Do they apply to the development of new antifungal agents?
Yes and no. Yes, in the sense that the GAIN Act that you mentioned, that was introduced in 2012, covered also fungal infections. Therefore, for example, our antifungal, ibrexafungerp, is covered by the GAIN Act, and it is a QIDP product, a qualified infectious disease product recognized by FDA. And therefore, we have some of the benefits of the GAIN Act. However, many recent organizations that have been supporting the development of new antimicrobials for pathogens that are resistant, organizations like BARDA or organizations like CARBEX, and they provided a significant support to companies, but companies developing antibacterials, not antifungals. This is changing, and probably because the emergence of these new pathogens that have been showing to be fungal pathogens, showing high aggressiveness, very difficult to treat, multi-drug resistance type of profile.
Why do you think we haven't seen more innovation around new antifungals?
Excellent question, Danny, because this is actually a question I get frequently. Why we don't have more antifungals? The reason is that it's very difficult to find proper targets against the fungi. The metabolic machinery of the fungi is not too different from the metabolic machinery of the human cells. So when you try to target something to kill the fungi, sometimes you are also causing toxicity to human cells. And whereas for bacteria are so different in terms of metabolic machinery from human cells that you can find many more ways to attack the bacteria without damaging the human cells. In fact, actually, there are two classes of antifungal, amphotericin B and the azoles, that have a mechanism of action that interferes also with human cells and that may cause some kind of toxicity that can, in the case of amphotericin B, can be significant. In fact, amphotericin B is always nicknamed by infectious disease specialists as amphoterrible because of a set of adverse events associated with this antifungal. So the reason of so few antifungals is because it's very difficult to find mechanisms that kill the fungi without harming the human cell. Our compound targets a structure that does not exist in human cells, and therefore the risk of off-target effects, toxic off-target effects, is much lower. And we have seen our drug to be extremely clean in terms of safety profile.
Well, your lead candidate is ibrexafungerp, the first of a new class of antifungals. What exactly is it?
It's a small molecule, and it targets a specific structure of a fungi. The cell of a fungi is surrounded by a hard shell. Think like the hard shell of the egg that protects the developing chick inside. Our product blocks the production of the building blocks, the glucan, that makes this hard shell around the fungi. By blocking this production, so this is why our product is called a glucan synthase inhibitor, the hard shell breaks down, and at that point, the cell just simply dies. And this hard shell around the cell is a structure that is not present in human cells. And this is why blocking and destroying this structure around the fungi cell does not have a negative effect on human cells.
How does this mechanism of action differ from the other classes of antifungals today?
Well, amphotericin B and azoles, which are the two first classes, and the azoles actually probably is the antifungal class used the most because it's the only one with orally bioavailable antifungals. They affect the metabolism of the fungi. And in fact, azoles, for example, they are fungistatic. They block the growth of the fungi, but they are not fungicidal. They don't kill the fungi. Our product, by blocking the production of this hard shell around the fungi, actually is fungicidal. It kills the fungi. There is another class of currently available antifungals, the echinocandins, that they target the same structure that we target, this hard shell made of glucan. But since the two classes, echinocandins and ibrexafungerp, are so different, fungi that have become resistant to echinocandin are still sensitive to our product because the two products, the echinocandins and ibrexafungerp, they attack the cell wall of the fungi in different ways.
Your lead indication is in vaginal yeast infections. What's known about the efficacy of the drug to date?
Well, very good. Danny, let me just say that actually that is our leading indication right now. We completed the phase 3 program in April, and we are now getting ready to file this year, in a few months, our first NDA. The product is a fast-track priority review product, and therefore we expect approval of the product mid of the next year. So we have shown with the vulvovaginal candidiasis program that our product is as activity, clinical activity, is well tolerated in patients, and therefore we have the best possible demonstration of the activity and of the product.
And why start with that as the first indication?
Well, actually, we started the development of the product for hospital infection. And in fact, we have a phase 3 program ongoing where we are testing ibrexafungerp in patients with invasive fungal infections. As I mentioned, these are cancer patients or transplanted patients with infections that are refractory to currently available antifungals. In other words, in patients who are not responding to currently available treatment. And we actually provided some very promising data in January based on an interim analysis of these patients. We have also a study against Candida auris and a study against pulmonary aspergillosis, which is also a very aggressive internal fungal infection of the lungs that actually have been associated recently to the use of ventilators in COVID-19 patients. So the program in hospital is moving along, is in phase 3. However, the program in vulvovaginal candidiasis, since there are millions of women suffering of vulvovaginal candidiasis, and unfortunately, the currently available treatment have significant gap in terms of efficacy and safety, made possible for us to do this program extremely quickly. So the vulvovaginal candidiasis program also started somewhat later than the hospital, actually leapfrogged the hospital program in terms of execution. We started the program, just to give you an idea, we started the phase 3 program last year, and we are going to file this year for the NDA. So the program was executed extremely fast.
How broadly applicable might this drug be?
Well, actually, like many antifungals, since there are so few of them, they are used usually in a broad range of indications, depending on the antifungal. Our antifungal, ibrexafungerp, has a broad spectrum of action. It works against Candida, Aspergillus, Pneumocystis, Blastomycosis, Histoplasmosis, and also Coccidioidomycosis. So it works against the vast majority of pathogens that can cause fungal infections. Therefore, we are developing the product in a broad range of indications that will go from community type of infection, like vulvovaginal candidiasis, and the prevention of vulvovaginal candidiasis recurrences, up to the deadly, high mortality, severe hospital infections in immunocompromised subjects, like, as I mentioned, pulmonary aspergillosis or invasive fungal infection refractory to other treatments. So it's a very broad range of indication that goes from the less severe infection, like vulvovaginal candidiasis, where we are using a low dose for 1 day. For vulvovaginal candidiasis, the treatment is a 1-day treatment, low dose, a 600 milligram given for 1 day. Whereas for hospital infection, where the infection is deep-seated inside the patients, we have to use a higher dose, 750 milligram, given sometimes for a few weeks or even for a couple of months in certain type of deep-seated type of fungal infections.
How big a concern is the potential development of resistance against the drug?
Well, let me just say, as an infectious disease specialist, I can tell you that sooner or later, pathogens, they always found a way to become resistant. The pathogens, microbes, have been around billions of years before us, and they will be around billions of years after us. The important point is that so far we have seen very little risk for development of resistance. So we expect, like for other antifungals, to have probably 15 to 20 years before resistance to a new antifungal really becomes a significant issue. For example, the quinocandins as a class, they were introduced in 2000, and it has been only a few years ago, around 2015, that we started to see in the clinics some fungi becoming resistant to quinocandin. So it took a good 15, 20 years before resistance became a problem. So we expect our drug that will be very effective against pathogens that are currently resistant to what is available today. Will one day developing resistance? Probably yes. That is the destiny of all the antimicrobials. And this is one important point I want to make, is that this is why it's so important to support the development of new anti-infectives products in general. Is because pathogens are very smart, are very, very smart. They find always a way to become resistant. And this is why it's so important to support the anti-infectives industry. Recently, we have seen some big companies like Merck, Pfizer, Eli Lilly that are realizing, and they made an announcement just a couple of weeks ago, that they are going to support the anti-infectives industry. It's important for our society that we continue to develop new products like ibrexafungerp to make sure that we have weapons that can fight the challenges of tomorrow.
Marco Taglietti, President and CEO of Scynexis. Marco, thanks so much for your time today.
Thank you very much, Danny. It was my pleasure to talk with you today.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.